So today we'll present one of the protocols, novel protocol used for preparation of bacterial ghost using a single chemical agent known as tween 80. Have you ever heard about bacterial ghost? I know it's a bit scary, but
It's a new terminology.
yeah.
It's new.
Anybody? It's new?
To me,
New
it's
for
new.
it.
Bacterial ghosts is new.
Yeah.
I know bacteria.
So, bacterial ghost is not a real ghost or human ghost. You know, it's bacterial ghost. What we mean by bacterial ghost? Bacterial ghost actually is an empty shell of bacteria evacuated from their contents. So for that reason we called it bacterial mummies or ghost. So they are evacuated and only we preserve and maintain the external shell of the bacteria in order to use or utilize these surface characters or bacterial characters only characters of walls and membranes. Actually, this definition of bacterial ghost has been extended to include, it was confined only in gram-negative until recently, until about 2013 or 2012. Before that, it was confined to gram-negative bacteria because it was used as gram-negative bacteria has a specific character or specific gene that's responsible for translation of a specific protein called E-lysis protein, which is responsible for evacuation or pore formation, one pore, single pore, which is used for evacuation of internal contents. Actually, this definition extended starting from 2012 to include gram-positive also. Although gram-positive lacking this E-lysis protein. So by using of some chemical agents that used for evacuation of gram-positive, this definition also includes gram-positive bacteria. So we can conclude that bacterial ghosts can be prepared by both ways. Genetically, using E-lysis gene, and as we mentioned, it's only confined to gram-negative bacteria. And chemically, can be used chemically by using of specific chemicals, specific agents at critical concentrations and critical times. And it can include both gram-positive and gram-negative bacteria. Actually, here is a list of some gram-negative and gram-positive bacteria that already prepared as ghosts within different periods of times. Escherichia coli, gram-negative, Salmonella typhimurium, Salmonella enteritis, Klebsiella pneumonia, Bordetella, Helicobacter, Vibrio, Haemophilus, Pasteurella, Pseudomonas, and so on. Some other gram-positive bacteria, and actually there are limited number of gram-positive bacteria that already prepared by chemical protocols, Staphylococcus aureus and Listeria. So gram-negative bacteria has a unique ability, only gram-negative bacteria has a unique ability to translate highly specific proteins. This protein is called E-lysis protein, which evolve transmembrane or can do transmembrane tunnel, which is responsible for evacuation. It's usually a single tunnel to evacuate the bacterial cell from their internal compounds or components. Under control processing, translation of this gene into lysis gene has been used to produce evacuated several gram-negative bacteria, as we mentioned in the last slide. This method was firstly used by a scholar, very common scholar, is called Lubitz. At 1984, this is the first trial to evacuate gram-negative bacteria using this E-lysis gene. The chemical way has been invented by another scholar called Amara in 2013 using multiple chemical agents and some controlled physical conditions through consecutive steps. This protocol was utilized in preparing Escherichia coli. We know Escherichia coli is gram-negative. Another trial at 2014 and 15 by Vinod, he prepared both gram-negative and gram-positive, Salmonella enteritidis gram-negative, and Staphylococcus aureus ghost as a gram-positive ghost for the first time by using a single chemical agent, which is sodium hydroxide. Our protocol, the current protocol, which invented by myself and my team, at 2017, this protocol was used for preparing of, the first trial was used for preparing of Salmonella typhimurium, Salmonella enterica serovar typhimurium. We have used one or single chemical agent, which is tween 80. Maybe some of us not familiar with tween 80. Tween 80 actually is a polymorphic chemical agent used as a surfactant and solubilizers, and used for specific purposes. For example, preparation of food, preparation of drugs, and used for solubilization. It's amphoteric components, has hydrophilic and hydrophobic parts, two parts, hydrophilic and hydrophobic. There are several types of tween 80, tween 20, and several compositions. We have used tween 80. We have used tween 80 as a single chemical agent, and after treatment by tween, pH was adjusted to 3.6 using lactic acid. Actually, this protocol has been approved and registered as a patent in Saudi Arabia, but we still in process. What about the application of bacterial ghost? Why we have to use, or why we are trying to prepare bacterial ghost? Actually, it is a biotechnology, new biotechnology platform, can be used and can be utilized in several applications. One of these applications, vaccination. We know that bacterial ghost, it's a dead bodies, so we can utilize it as a vaccine. You know, vaccines can be killed organism, can be attenuated organism, can be part of organism. So we use total bacterial shell. So we utilize the bacterial surface characters in order to stimulate immune system. The immune system will define this surface characters, and this is actually have been used and applied in several applications in both human and veterinary. Also, it can be used as targeted drug delivery system, as applied by by Werner Lubitz. He utilized E. coli ghost in order to target colon cancer. We know that E. coli has very strong affinity to colon cells. So by virtue of the surface character of E. coli and good affinity and strong affinity to colon cells, it will be targeted by these cells. And once we upload anti-cancer inside these shells or these bacterial ghosts, it can be target only colon cancer and avoid side effects and distribution of anti-cancer to other tissues. So we can overcome the side effects of anti-cancer. Actually, it can be used as DNA carriers. It's a carrier, actually. It's empty carriers. So you can upload everything. You can upload DNA, drugs, and also can be used in future in gene therapy. So bacterial ghost have several applications, and we can use it in different areas. The protocol, we have used one of the standard strains called Salmonella enterica cerovar typhimurium, American culture type, by specific code. Actually, before starting the protocol, we define or determine the MIC or minimum inhibitory concentration for some agents. For example, we have used tween 80, 5% SDS, and potassium hydroxide, sodium oxide, H2O2, benzoic acid, and editan and lactic acid. Tenfold dilutions were prepared for each reagent in order to determine MIC or minimum inhibitory concentration. And after that, aseptically, we have added 100 microliter of standard inoculum of Salmonella typhimurium, standardized by 0.5 Macfarland. Microbiologists know that. Added to each dilution, all tubes were incubated for 24 hours, then tested for turbidity. The tube lacking turbidity or clear tubes considered as the first dilution, lacking of turbidity considered as the MIC or minimum inhibitory concentration. The third group was treated after first incubation by addition of lactic acid until pH 3.6 and incubation for different periods of time, 20, 30 minutes and 1 hour, 2 hours and 3 hours. Every experiment was triplicated, then all results were taken in average. After that, to obtain pure cells, to obtain only pure cells, whatever killed or live, we have done centrifugation. The supernatant was utilized for quantification of proteins and DNA. The supernatant used for quantification of DNA and proteins, and the precipitate pellets were washed by sterile normal saline 3 times. In order to analyze proteins and DNA we have used a specific protocol called Bradford protocol for analysis of protein. It's a very famous protocol. And using a specific device called nanodrop device in order to quantify proteins in addition to DNA. All results were taken at 595 nanometers. In the same way, we have used the same device, nanodrop, to quantify DNA quantities. The centrifuged pellets were investigated in order to investigate the pores the surface pores by electron microscope, by scan electron microscope. The DNA extracted also was analyzed using electrophoresis, gel electrophoresis. And also we have confirmed, excuse me, confirmed the integrity of the external surface of cells by gram staining and light microscope. Light microscope and gram staining in order to confirm the integrity of the surface of cells. To confirm that cells are not degraded. Okay? In order to preserve bacterial ghosts for a long time, we have lyophilized it. Lyophilization, you know, using of freezing and drying at the same time. And we have done statistical analysis using ANOVA analysis. Several trials have been done within several months in order to optimize the results. This is a flow chart of the whole protocol. I can summarize this protocol. Incubation of cells, of target cells, either gram positive or gram negative, with 7% tween 80, including rich media. In rich media, for example, molar Hinton, we have used molar Hinton media, agar, for 24 hours at 37 degrees. Addition of lactic acid to reach 3.6 and standing for 1 hour at 37. Centrifugation for 10 minutes. And discarding supernatant and washing by half normal saline. Then repeat centrifugation and washing 2 times, extra 2 times. And subculture pellets to confirm full killing. This is a very important step in order to confirm killing of the whole population. The other protocols or other ways for preparing bacterial ghost usually obtain about 90, 99, 95% of death, of killing of population, which is a bit dangerous when we are using it as vaccine. We have some live organism. So it's important, fully important, it's very important to kill 100% population. So in order to confirm the safety of the product, we have to use, or we have to subculture in order to confirm the full killing. After that, lyophilization and store at room temperature. The MICs of different components, different agents like potassium hydroxide, sodium hydroxide, and others about these values. And for hydrogen peroxide, SDS, and tween 80 were not determined because all of these dilutions, all dilutions of H2O2 or hydrogen peroxide inhibits all growth, all dilutions. So MIC was not determined. The others, like tween 80 and SDS, didn't inhibit actually any growth. So there is no MIC. But tween 80, although all concentrations will not inhibit any growth, but we have seen that tween 80 achieved the best results. How? None of the used reagents produce high-quality bacterial ghosts when added to the culture broth after incubation of 24 hours, except tween 80. All of these agents didn't give any good results except tween 80. Incubation of Salmonella typhimurium in molar Hinton broth containing 5% of tween 80 result in cell perforation. And all used reagents other than tween 80 has produced cells which lacking the integrity of their external characters. So other agents kill all organisms, okay, but without preserving their integrity or their surface characters. They disform the external characters, except tween. Although tween non-killer or were not kill any of the organism, but still perforate some cells without full killing. In order to solve this problem, we have used another factor, which is pH. So by sudden reducing of pH to 3.6, all cells were killed. This is the electron microscope figure that illustrate the several pores. This is before treatment. These cells are bacillus, Salmonella, typhimurium bacillus. All are integer without treatment, no pores. Here some pores after treatment of tween. We can notice some pores, some surface pores induced by after treatment with tween. Here are some surface pores. So after that, we have to also to quantify the quantities of proteins and DNA in order to confirm the evacuation of cells. So incubation of cells with tween 80 at 5%, 6%, and 7% concentrations lead to variation in proteins released in DNA. We have noticed that the highest quantities of DNA of proteins were released at 7%. The highest quantities of DNA released at 6% of tween. And this is the optimum condition that has been chosen to prepare the bacterial ghost. At 1 hour, we can see that the highest quantities of DNA after incubation with tween 80 for 24 hours. And after half an hour, we have obtained the highest amount of DNA, released DNA. The subculture of isolated cells was positive, which indicates incomplete ghost preparation. We cannot consider complete high-quality bacterial ghosts in case of presence of some live bacteria. So we have to, as we mentioned now, we have to obtain 100% killing. So using different trials, using, for example, or different experiments, tween only give us release protein about 507. Tween with freezing, extra treatment or manipulation in order to kill all population, we have obtained 595. Tween 80 with lactic acid achieved the largest amount of protein. In case of DNA, tween 80 almost null. Also with freezing 1.5, which is very little. By using of tween with lactic acid, it achieved very high amount of DNA released. By electron microscope investigation, using tween, we have obtained numerous pores. By using of freezing pores more. Using of tween plus lactic acid, we have obtained very large amount of pores. By using of light microscope, the integrity of cells are positive in all. All cells are integer and not deformed. By subculturing and confirming safety of product, tween 80 has subculture growth, which is non-safe, and also tween 80 plus freezing also are positive, but by using of tween 80 plus lactic acid, there is no growth after subculturing. It means it was safe product. So in order to optimize bacterial ghost quality, how can we know bacterial ghost with high quality? The high quality bacterial ghost means that bacterial ghost preserving full surface characters. So it it looks like a live bacteria, like human ghost, like scientific fiction talking about ghosts. Ghost is about, it looks like a human body, full human body, but internally like zombies or something like that. They are dead, but full body. Also, bacterial ghosts quality with high quality, full and complete surface characters, bacterial cells without internal contents, evacuated shells. So, in order to optimize the bacterial ghost quality, means that only bacterial shells with evacuated substance and without deformation of surface characters, we consider it as high quality bacterial ghost. In order to optimize the quality, different periods of exposure to lactic acid were tested. As I mentioned, 20 minutes, 30 minutes, 1 hour, 2 hours, and 3 hours. The highest amount of quantities of proteins and DNA were at 1 hour and half hour respectively. So we have chosen the 1 hour. Gel electrophoresis analysis, DNA was extracted from the pellets of untreated and treated cells, showing difference in bands intensity. When we compare the different bands, this is a control without any treatment. We can notice that the full amount of DNA in comparison with the using of tween only is comparable to control. The tween with freezing comparable with or less, the quantities were less than the control and tween only. The using of tween plus lactic acid, we have noticed that very little amount of DNA. It means that DNA were lost after treatment of tween and lactic acid. So, in conclusion, the optimum treatment for producing the desired highest quality bacterial ghost, killed and perfect cellular shells, was achieved by incubation of bacterial cells with 7% tween 80 for 24 hours and reducing pH to 3.6 by adding lactic acid for 1 hour. So this is the optimum condition for preparing the bacterial ghost. So in comparison to other protocols, for example, genetic method, which is used, as we mentioned, confined using for only gram-negative bacteria, in addition to the sophistication of this method, and in addition also to the probability of production of some live bacteria, which is dangerous. It will be not safe in case of using of anti-cancer or drug delivery system for anti-cancer or using of vaccine. So this is one of the dangerous drawbacks of this method. The chemical agents can be used in critical concentrations and critical times in preparing both gram-positive and gram-negative. Recently, actually, in current year and in 1 year ago, there are several trials in preparing yeast or fungus ghosts and even viral ghosts. Gram-positive bacterial ghosts can be chemically obtained by the same concepts that are used for preparing of gram-negative. High-quality ghosts have been obtained by using suitable chemicals, optimizing the concentration, temperature, and shaking speed. Why we have used some chemical agents? Chemical agents were chosen because for their impact on their outer surface, of the bacteria or bacterial cells or bacterial membranes. The permeabilization effect of the outer membrane or on the cell walls is the common effective factor of these agents. In the previous protocols, for example, using of sodium hydroxide as a single agent used for preparing of both gram-positive and gram-negative, for example, Vibrio cholera or Parahemolytica and Salmonella enteritidis, and Staphylococcus aureus. But sodium hydroxide has very drastic effect and destructive effect on the external shell. And this is actually breach the quality or the high quality of bacterial ghost. So we cannot use sodium hydroxide, even though some of colleagues use it, and actually his trial was successful. Alkanes have damaging effect on the outer membrane, so it's usually deforming the external surface. Sodium dodecyl sulfate (SDS) is an amphoteric compound which has hydrophilic and hydrophobic tail, so it affects the outer membrane and permeability of the outer surface. Hydrogen peroxide oxides essential external cellular components of the by producing free radicals. Accordingly, tween 80 was the sole key point, the key player in this protocol. High quality, perfect, three-dimensional bacterial shell was achieved using different concentrations of tween 80 alone without inhibition of growth or killing of the whole population. So in order to solve this problem, we have used the lactic acid in order to kill whole the population. Actually, why we have chosen the tween 80? Tween 80 is a non-anionic surfactant used for food and drug industry and used for solubilization of hydrophobic components. So by using of tween 80, we can dissolve the external hydrophobic components of the bacterial cells. It was used in Pseudomonas aeruginosa, gram-negative, and the longer exposure of tween 80, the higher degree of leakage of outer components. It was obvious that tween 80 in concentrations 5–6% was very effective in bacterial activity of number of antibiotics. This is previous studies. The release of reasonable quantities of proteins and DNA is a strong indicator for effective preparation of ghost. In our protocol, the maximum liberated quantities of proteins and DNA were 2975 microgram per ml and 793 microgram per ml respectively for DNA and proteins. In other protocols, preparing, for example, E. coli, the highest amount of proteins released about 3425 and the DNA released 179. Actually, the full antigenic characters of obtained ghosts need further investigation by observing their ability to elicit a valuable immune response. Actually, we have done this and we got very amazing results, but still unpublished. I cannot propose it. So, and the application of this protocol or in preparation of bacterial ghosts, gram-positive bacteria should be examined, still in process. But we have applied it in immunology, in immunological preparations as a vaccine. We got amazing results, and also we have used it in preparation of drug delivery system as anti-cancer, still also in process, and we got amazing results, but unpublished results. This work has been published in Saudi Pharmaceutical Journal in February 2018, and we are very grateful for Kayali Chair for Pharmaceutical Industry, which is related to College of Pharmacy King Saud University, for funding this work. In conclusion, the current protocol, conclusion should be before this. The current protocol of chemically induced ghost preparation can be used for obtaining high quality and surface integrated ghosts of Salmonella typhimurium. It's cheap, simple, feasible, and avoids the complications of genetic approach. Further investigation is needed to study their utilization as drug delivery system and vaccines. That's all. Thank you very much. If you have any question.
Thank you for a nice presentation of a new science.
Thank you.
And hope that it will help to eradicate that colon cancer in future. It's a serious issue.
Yeah, that's right.
Now, tween 80 is a liquid, right? It's a liquid.
Yeah.
And it is used in agriculture also as a surfactant.
Yeah.
Right? What prompted you to use this in this subject? Did you get some information, literature that it could be used in medical science also or pharmaceuticals?
Yeah.
Because you know that the industry of insecticide, it is largely used.
Yeah.
So that the spray doesn't fall down of the leaves, particularly those leaves which are smooth and cannot withstand the droplets. So what prompted you to think about it? Any literature on that you got it?
Yeah, a lot of literature talked about tween 80 used as we mentioned, it was used with Pseudomonas aeruginosa. It has it was very effective in enhancing the effect of antibiotics that use against Pseudomonas aeruginosa. A lot of literature talked about tween in drug industry and antimicrobials. I can supply you with some of these literature. Sure.
Okay, some of the detergents, simple detergents that we use and available in the supermarkets, do you feel that such detergents can serve this purpose of tween 80? Do you think that that may be useful also?
There are several detergents, you are right, but you know, we have done a lot of trials, including detergents, alkalis, and so on. All of detergents and alkalis that we have chosen and that we have tried didn't give any good results. Most of them destroy the surface, whole surface. So this, you know, breach the concept of high quality bacterial ghosts.
Thank you.
Yeah, okay.
I want to ask if the cell envelope has a lifetime, or another question, does the cell envelope contain enzymes make autolysis of it? Also, the identification of bacteria, may the ghost bacteria can be identified like the real bacteria, the different species?
Okay. Regarding to the first question, I mentioned that we lyophilized. You know, lyophilization is one of the preservation methods, using drying and freezing at the same time. So most of lyophilized, even live bacteria, lyophilized live bacteria the lifespan about more than 10 years. So we can preserve it at room temperature for 10 years. Actually, stability study, this is recently prepared. So stability study needs long time, you know. But theoretically, lyophilization can preserve product for very long time. The second question, which is about the lysis, E-lysis or lysis proteins. Lysis proteins only exist in gram-negative bacteria and in live gram-negative bacteria. So this is one of the drawbacks of using genetic method, the genetic protocol in preparing bacterial ghosts. We cannot use the same concept in gram-positive. After killing of bacteria, there is no E-lysis gene. Regarding the third question, identification. There is no need for identification of ghosts, because I have prepared gram-negative ghosts and already identified. So no need for identification. Okay.
Any more questions?
Thank you very much, Dr. Sameh, for sharing this
You are welcome.
cutting-edge technology.
I appreciate it. Thank
It's
you.
absolutely new to me.
Thank
and
you.
I believe in this, or this house as well.
So a bit scary, yeah?
Yeah. So, and from your presentation, we have noticed that this bacterial ghost has the multiple use, including the the vaccination, the gene therapy, and for the colon cancer as well.
Yeah.
So this can be a milestone in the medical science
That's right.
in the world. Yeah. So, so who is getting the patent right? It's you or the Saudi government or who
No.
else?
It's myself and our team. This is my team. The head of KLA chair. It's a research chair related to contributed to pharmacy college in King Saud University. This is my team. Actually, the patent will be involving myself and Dr. Munir, Dr. Faris, all these three, because all of us living in Saudi Arabia. The other professors, these are my professors, Dr. Ayman, Dr. Naira, Dr. Abdul Jawad. All of these professors are living in Egypt. So, and for administrative issues, the patent will be involve myself, Dr. Munir, and Dr. Faris, the first three guys.
And King Saud University is a public university or private?
King Saud?
Yeah.
Yeah, King Saud University, it's under umbrella of King Saud University. Actually, the patent will be released to Al-Kayali chair for those three persons.
Okay, thank you.
You're welcome. Any more questions?
Any more questions? Okay. Okay. Thank you, Dr. Rabea, for this wonderful talk.
Thank you very much.
So may I call upon Dr. Jay Barnard for his talk?